Hello, and welcome to your Microbiology Bio 203 video lecture on microbes in our lives. I'm Mr. Kennedy, and I will be your guide as we explore this topic together. Microbes are organisms that are too small to be seen with the unaided eye. Microbes include bacteria, fungi, protozoa, microscopic algae, and viruses. Contrary to popular belief, few microorganisms are actually pathogenic or disease-producing. Most are beneficial. They decompose organic waste, generate oxygen by photosynthesis, produce chemical products such as ethanol, acetone, and vitamins, produce fermented foods such as vinegar, cheese, and bread, and potentially produce products used in manufacturing and disease treatment. Designer genes have their origins deeply rooted in the world of microbiology. Everything from denim fading to the plastics in the fabric have microbial origins. Microbes in our lives. It's essential for everyone to have a working knowledge of microorganisms, regardless of their future careers. Knowledge of microorganisms allows humans to prevent food spoilage, to prevent disease, and to understand causes and transmission of disease to prevent epidemics. Naming and classifying organisms. Carolus Linnaeus established the current system of scientific nomenclature in 1735. Carolus Linnaeus is also known as Carl von Linné, and he created the two-name naming system currently referred to as binomial nomenclature. In today's naming scheme, all organisms receive two names, the genus and the specific epithet. When written correctly, scientific names are italicized or underlined. The genus is capitalized, the specific epithet is lowercase. Scientific names are Latinized and used worldwide. They may be descriptive or honor a scientist or some combination of the two. Escherichia coli is a good example of such a case. Escherichia honors the discoverer Theodore Escherichia and describes the bacterium's habitat, the large intestine or colon. Staphylococcus aureus describes the clustered spherical coccal cells and describes the golden-colored aureus colonies. After the first use, the scientific name may be abbreviated with the first letter of the genus and the specific epithet. As an example, Escherichia coli and Staphylococcus aureus are found in the human body. In their second use, E. coli is found in the large intestine, and S. aureus is on the skin. Types of microbes: bacteria, archaea, fungi, protozoa, algae, viruses, multicellular animal parasites. Bacteria. Bacteria are prokaryotes. The word prokaryote literally translates to pre-nucleus, as these organisms lack a nucleus and any membrane-bound organelles. Bacteria are single-celled, have peptidoglycan cell walls, divide via binary fission, and derive nutrition from organic or inorganic chemicals or photosynthesis. The archaea are also prokaryotes. They lack peptidoglycan cell walls, often live in extreme environments, and include the methanogens, extreme halophiles, and extreme thermophiles. The fungi are eukaryotic organisms and are distinguished from prokaryotes by the presence of a distinct nucleus and a variety of other membrane-bound organelles. Fungi have chitin in their cell walls. They absorb organic chemicals for energy. Yeast is an example of a unicellular fungus, while mold and mushrooms are multicellular. Molds consist of masses of mycelia, which are composed of filaments called hyphae. Protozoa are also eukaryotes. They absorb and ingest organic chemicals. They may be motile via pseudopods, cilia, or flagella. They can be free-living or parasitic. As a parasite, they would derive nutrients from a living host. Algae are eukaryotes. They have cellulose cell walls, can be found in freshwater, saltwater, and soil. They use photosynthesis for energy and produce oxygen and carbohydrates. Viruses are acellular. They consist of DNA or RNA cores. The core is surrounded by a protein coat, and the coat may be enclosed in a lipid envelope. Viruses are able to replicate only when they are in a living host cell. Outside of a host cell, they are inert and may remain so for hundreds of years. Multicellular animal parasites are all eukaryotes. They are multicellular animals and not strictly microorganisms. Parasitic flatworms and roundworms are called helminths and make up the majority of these individuals. Some helminths have microscopic stages in their life cycles. Classification of microbes. The current classification scheme organizing microbial life into 3 domains was developed by Carl Weiss. The 3 domains are based on cellular organization and are as follows: the bacteria, the archaea, and the eukarya. The eukarya is subdivided into the protists, fungi, plants, and animals. This graphic will help you understand the organization of the 3-domain system. Brief history of microbiology. The first observations of microbes were done in 1665 by a man named Robert Hooke. Robert Hooke reported that living things are composed of little boxes or cells. This marked the beginning of cell theory. All living things are composed of cells. The first microbes were observed from 1623 to 1673 by Anton von Leeuwenhoek. Von Leeuwenhoek described animalcules viewed through a magnifying lens. Upon the discovery of microbes, the debate over spontaneous generation began. Spontaneous generation is the hypothesis that life arises from non-living matter or a vital force is necessary for life. Biogenesis is the hypothesis that living cells arise only from pre-existing living cells. This debate was tested in laboratories by a number of scientists. In 1668, Francisco Redi filled jars with decaying meat to determine whether or not flies came from other flies or from decaying meat. In his experimental design, meat was placed in jars. Some of the jars had a burlap lid, while others had no lid at all. The jars that were covered with a fine net had no maggots on the meat. However, there were maggots on the net. The open jars had maggots all over the meat. Francisco's Redi experimental design proved that spontaneous generation was false for large organisms such as insects. In 1745, John Needham boiled beef broth and put it in covered flasks to attempt to disprove spontaneous generation in a slightly different way. His nutrient broth was heated and placed in a covered flask that was capped with a cork. Once set out on the lab bench, it spoiled within a number of hours. John Needham hypothesized that spontaneous generation was actually real as a result of his laboratory results. In 1765, Lazzaro Spallanzani repeated John Needham's experiment with one small modification. Rather than using a cork to seal his flask, Spallanzani literally melted the glass end of the flask shut. Once the beef broth was boiled and the flask was sealed, the broth remained unspoiled for days, weeks, and months. Therefore, as a result of Spallanzani's work, the idea of spontaneous generation was put down as long as air was not allowed to make contact with the broth. Out of this idea came the hypothesis that there was a vital force in the air that imparted life to the beef broth, and as long as that air was not allowed to touch the broth, the vital force could not create life. The theory of biogenesis was first drafted in 1858 by Rudolf Virchow, who said cells must arise from pre-existing cells. In 1861, Louis Pasteur tested this idea. Louis Pasteur demonstrated that there were microorganisms in the air, and it was microorganisms that was causing broth to spoil in John Needham's experiment, not a vital force or mythological power. In Louis Pasteur's experiment, he took nutrient broth, heated it, placed it in a flask, and left it unsealed, and the broth spoiled. He then took nutrient broth, placed it in a flask, and then immediately sealed it, and there was no microbial growth. Pasteur also used a renowned S-shaped flask in his experimental design. The S-shaped flask was designed to keep microbes out but to let air in. Broth in the flasks with the long S-curved neck showed no signs of life. The neck of the flask trapped the microbes, thus proving that microbes originate in the air or fluids, not mystical forces. This is an illustration of Louis Pasteur's work using his S-shaped flask. Brief history of microbiology. The golden age of microbiology occurred between 1857 and 1914. Beginning with Pasteur's work, discoveries included the relationship between microbes and disease, immunity, and antimicrobial drugs. Pasteur showed that microbes are responsible for fermentation. Fermentation is the microbial conversion of sugar to alcohol in the absence of air. Microbial growth is also responsible for spoilage of food and beverages. Bacteria that use air spoil wine, turning it into vinegar or acetic acid. Pasteur demonstrated that these spoilage bacteria could be killed by heat that was not hot enough to evaporate the alcohol in wine. Hence, pasteurization was born. Pasteurization is the application of a high heat for a short time to kill harmful bacteria in beverages. Louis Pasteur was alive and working in the field of microbiology from 1822 to 1895 and is most well known as being the person who demonstrated that life did not arise spontaneously from non-living matter. Germ theory of disease. In 1835, Agostino Bassi showed that a silkworm disease was caused by a fungus. Louis Pasteur in 1865 showed that another silkworm disease was caused by a protozoan. In 1840, Engels Semmelweis advocated hand washing to prevent transmission of puerperal fever from one obstetrical patient to another. In the 1860s, applying Pasteur's work showing that microbes are in the air, can spoil food, and cause animal diseases, Joseph Lister used a chemical antiseptic, phenol, to prevent surgical wound infections. Joseph Lister was alive and working in the field of medicine from 1827 to 1912 and is noted as being the first to perform surgery under aseptic conditions using phenol and prove that microbes caused surgical wound infections. In 1876, Robert Koch discovered that a bacterium causes anthrax and provided the experimental steps, now known as Koch's postulates, to demonstrate that a specific microbe causes a specific disease. Robert Koch was alive and working from 1843 to 1910 and established the experimental steps for directly linking a specific microbe to a specific disease. In 1796, Edward Jenner inoculated a person with cowpox who was then immune from smallpox. This was the first attempt at vaccination. Vaccination is derived from the Latin word vacca, meaning cow. The protection is called immunity. The first synthetic drugs, quinine, were made from tree bark, which was long used to treat malaria. Paul Ehrlich speculated about a magic bullet that could destroy a pathogen without harming the host, and in 1910, Ehrlich developed a synthetic arsenic drug, salvarsan, to treat syphilis. In the 1930s, sulfonamides were synthesized. In 1928, Alexander Fleming discovered the first antibiotic, literally by accident. Fleming observed that Penicillium fungus made an antibiotic, penicillin, that attacked S. aureus. In the 1940s, penicillin was tested clinically and mass-produced. Modern developments in microbiology. Bacteriology, mycology, and parasitology are growing fields of microbiology. Bacteriology is the study of bacteria, mycology is the study of fungi, and parasitology is the study of protozoa and parasitic worms. Immunology is the study of immunity. Vaccines and interferons are used to prevent and cure viral diseases. A major advance in immunology occurred in 1933 when Rebecca Lansfield classified streptococci based on their cell wall components. Virology is the study of viruses. Dmitry Ivanowski in 1892 and Wendell Stanley in 1935 discovered the cause of mosaic disease of tobacco as a virus. Electron microscopes have made it possible to study the structure of viruses in detail. Recombinant DNA technology. Recombinant DNA technology opens a door to the world of microbial genetics. Microbial genetics is the study of how microbes inherit traits. Molecular microbiology is the study of how DNA directs protein synthesis. Genomics is the study of an organism's genes and has provided new tools for classifying microorganisms. Recombinant DNA is DNA made from two different sources. In the 1960s, Paul Berg inserted animal DNA into bacterial DNA, and the bacteria produced an animal protein. In 1941, George Beadle and Edward Tatum showed that genes encode a cell's enzymes. In 1944, Oswald Avery, Colin McLeod, and Macklin McCarty showed that DNA is the hereditary material. In 1953, James Watson and Francis Crick proposed a model of DNA structure. And in 1961, Francois Jacob and Jacques Monod discovered the role of mRNA in protein synthesis. This is a timeline of their work in the golden age of microbiology. Microbes and human welfare. Microbes play a number of roles in human welfare. Microbial ecology is the study of the relationship between microbes and their environment. Microbes can convert carbon, oxygen, nitrogen, sulfur, and phosphorus into forms that are used by plants and necessary for animal life and survival. Bioremediation. Bioremediation is using microbes to clean up pollutants. Bacteria can degrade organic matter in sewage. Bacteria can degrade or detoxify pollutants such as oil and mercury. Insect pest control by microbes. Microbes that are pathogenic to insects are alternatives to chemical pesticides. They can prevent insect damage to agricultural crops and disease transmission. One such organism is Bacillus thuringiensis, which has infections that are fatal in many insects but harmless to animals and plants. Modern biotechnology and recombinant DNA technology. Biotechnology is the use of microbes for practical applications, such as producing food and chemicals. Recombinant DNA technology enables bacteria and fungi to produce a variety of proteins, vaccines, and enzymes. Missing or defective genes in human cells can be replaced using gene therapy. Genetically modified bacteria are used to protect crops from insects and from freezing. Microbes and human disease. Humans possess what is often referred to as a complement of normal microbiota, bacteria that should and do exist on a human and in a human in a positive symbiotic relationship. Bacteria were once classified as plants, giving rise to the term flora for microbes. This term has been replaced by the word microbiota. Microbes normally present in and on a human are commonly referred to as normal microbiota. Your normal microbiota prevent the growth of pathogens. Normal microbiota produce growth factors such as vitamins B and K. Resistance is the ability of the body to ward off disease, and it is increased through your normal microbiota. Resistance factors also include your skin, your stomach acid, and antimicrobial chemicals that the body produces naturally. Biofilms. Microbes can attach to solid surfaces and grow in mass. They will grow on rocks, pipes, teeth, and medical implants. Biofilms can cause infection and are often resistant to antibiotics. Emerging infectious disease. When a pathogen invades a host and overcomes the host's resistance, disease ensues. Emerging infectious diseases, or EIDs, occur when new diseases and diseases that are prevalent increase in incidence. Middle East Respiratory Syndrome, MERS, is an example of an emerging infectious disease. It is caused by Middle Eastern Respiratory Syndrome coronavirus, or MERS-CoV, common to SARS, and SARS can create severe acute respiratory syndrome. Middle East Respiratory Syndrome, or MERS, accounts for 100 deaths in the Middle East from 2012 to 2014. Avian influenza A, also known as H5N1, is caused by influenza A virus, which is primarily in waterfowl and poultry. It's sustained human-to-human transmission and has not yet occurred. Methicillin-resistant Staphylococcus aureus, or MRSA. Methicillin-resistant Staphylococcus aureus has come and gone over the past several decades. In the 1950s, penicillin resistance developed. In the 1980s, methicillin resistance developed. In the 1990s, MRSA resistance to vancomycin was reported. VISA is vancomycin intermediate S. aureus, and VRSA is vancomycin resistant S. aureus. West Nile encephalitis, or WNE, is caused by West Nile virus. It was first diagnosed in the West Nile region of Uganda in 1937. It appeared in New York City in 1999. In non-migratory birds, it exists in 48 states. It can be transmitted between birds and to horses and humans by mosquitoes. Bovine spongiform encephalopathy is caused by a prion, an infectious protein that also causes Creutzfeldt-Jakob syndrome. There is a new variant of CJD in humans, and it is related to cattle that have been given feed made from prion-infected sheep. E. coli. E. coli, subspecies O157H7, are a toxin-producing strain of E. coli. It was first seen in 1982 and causes bloody diarrhea. It is the leading cause of diarrhea worldwide. Ebola hemorrhagic fever, or EHF, is caused by the Ebola virus. It causes fever, hemorrhaging, and blood clotting, and is transmitted via contact with infected blood or body fluids. It was first identified near Ebola River in the Congo. 2014, there was an outbreak that occurred in Guinea and killed hundreds. Cryptosporidosis is caused by Cryptosporidium protozoa. It was first reported in 1976. Cryptosporidosis causes 30% of diarrheal illness in developing countries, and in the United States, it is transmitted via water. Finally, AIDS, acquired immunodeficiency syndrome, which is caused by human immunodeficiency virus or HIV. It was first identified in 1981. It is a sexually transmitted infection affecting males and females alike. Worldwide, uh, it has infected 35 million people with approximately 6,000 new infections every day. HIV, or AIDS, in the United States, statistics include 26% of patients who have the virus are female and 49% are African American. This concludes our coverage of microbes in our world.